Okay, good afternoon, everyone. So we will start this embryology session. Last, if you remember what we have done, I'll just go back quickly. What we have studied, we have discussed about fertilization topic, and in this topic we had studied different sections. That is approximation of the gamete. Can you all hear me?
Yes, ma'am.
Yes, ma'am.
Okay. So approximation of the gametes. Then we studied what is the structure of secondary oocyte, how the transportation of male gametes takes place in the reproductive tracts, and how during ovulation what kind of changes happens in the female genitalial tract, and the sperm count and analysis. Then the next comes maturation of sperms and different steps we had gone through. I'm sure if you can remember those steps. If not, just go back and see the recordings of those videos, what we have done in the last sessions, because that will serve one of our most important question, that is fertilization. Then the third step, that is crossing of the three barriers. The three barriers are the corona radiata, zona pellucida, and the cell membrane of the oocyte. What kind of changes we had seen? One more beautiful video of fertilization. And lastly, in fertilization, we have talked about effects of fertilization, right? So we know that what are the main effects of fertilization? Let us just quickly go through. That is, secondary oocyte is stimulated to complete the second meiotic division. This second meiotic division is completed only after fertilization takes place. Then the normal diploid number of chromosomes is restored, because egg has got, oocyte has got 23 number of chromosomes, and sperm has got 23 number of chromosomes. During fertilization process, when these two meet, female and male pronucleus meet, we are going to again get restored the total number of diploid chromosomes, that is 46. Next is variation of human species is happening, that is maternal and paternal chromosomes, they are intermingling with each other, so totally giving rise to entire new pattern of chromosomal genetics, which we will see in a new individual. Then also we will see that the embryo contains only maternal mitochondria, because the sperm mitochondria are dispersed into the egg cytoplasm and discarded. Also determination of the sex of the embryo. So this, that is because of diploid number of chromosomes, total 46 number of chromosomes are present. Out of that, the last pair are the sex chromosomes. It is X and X. It results in the formation of the female individual. If it is X and Y, it results in the formation of male individual, right? So these are the results of fertilization. So we also had discussed various contraceptive methods and how to follow them, and what is the composition of various contraceptive procedures which are available today in the market. So mechanism of action we had discussed in the last class. So that's all about the last session. Now we will move on to the first week of development. First week of development is from the time of fertilization to the implantation. Okay, implantation. What is implantation? We'll just go through quickly after completing the stages. So what is the first day of fertilization? After the first day of fertilization, what is actually happening? The first day, how, from where we are going to start is from the moment where the fertilization has happened. From that moment onwards, we are counting it as first day of gestation, first day of fertilization. Okay, so that is where we start our first week. Okay, in first week, what are the changes happening? We start with cleavage division. What is cleavage division? There is a video again to show what is cleavage division. This is the ampulla of the fallopian tube. You can see this is still the egg, and where fertilization has happened, and then this is now called as zygote. Slowly the zygote is moving towards the uterus. This is what you have to observe. Out of that, the zona pellucida is still intact. Okay, what layers? We have seen that there are 3 layers the sperm crosses, right? Corona radiata, zona pellucida, and oocyte ka cell membrane. Out of that, all the corona radiata cells are removed off by whatever extra sperms are there. They keep on removing this corona radiata cells, and finally corona radiata cells are free here. Okay, it's totally removed off. Then comes the second layer, that is zona pellucida. Zona pellucida is a very important structure. When it will be removed off? It will be removed off during implantation process. So zona pellucida is a very important question here. We will discuss that once we are talking about implantation. So what is important here is zona pellucida still remains intact. Now the newly formed cell, a single cell, still a single cell, where egg and sperm fuses, male and female pronuclear fuses to form a single cell stage. The single cell stage is still existing, for very few very little time, we have a single cell stage. If you see your one of your book, that is Inderbir Singh, it says that there is no single cell stage. Cleavage division starts immediately once the fertilization is ending up. Then itself the cleavage furrow starts and divides into 2 cells. But if you see books like Langman and Gray's Anatomy, which is the 32nd edition, they have corrected this stage that there do exist a single cell stage, and this single cell stage is for a very brief seconds, there is a single cell stage. And after this, what happens immediately? The single cell stage shows a cleavage furrow. Cleavage furrow means there is a depression presenting between inside the cell and from outside. And then immediately it divides into 2 different cells. In these 2 different cells, if you see, you will see that one cell is bigger and the other cell is smaller. Okay, why does it happen that way? Why does it happen that way? There is no specific reason why it happens that way, but it the division just happens, and during this division one cell remains larger and other cell remains smaller. Okay, this is called as cleavage division. Okay, cleavage division. So cleavage division starts, and immediately the larger cell starts dividing. Again a 2 cell stage. Now the 2 cell becomes 3 cell, 3 cell becomes 4 cell within 40 hours post fertilization. Okay, these all cells are dividing and accumulating inside the zona pellucida. You can see that zona pellucida is still intact. Now this is the stage. Okay, sorry. You can see that. We'll quickly go to this single cell stage, zygote, which is called as zygote. Now the zygote gives cleavage division, which leads to 2 cell stage, a large cell and a smaller cell. A large cell divides into another cell. We get now 3 cell stage, then immediately 4 cell stage. Okay, 4 cells divides to give rise to 8 cell stage. This 8 cell stage is called as morula. What is it called as? Morula. I hope you are all understanding this. Reply. Guys, can you understand this?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yeah, please
Yes,
concentrate
ma'am.
and listen. If you have any doubts, if you feel that you have missed some steps, please let me know so that I can revise it again. Okay. So you can see a 8-cell stage which is called as the next stage, which is called as morula. Okay, before morula we see blastula. Okay, what is it? Blastula. And after blastula, the next stage is morula. Okay, morula, morula. Till here, everyone clear?
Yes, ma'am.
Right.
Yes, ma'am.
So now what is happening here? Slowly, you can see that the cells are dividing again and again. This is 8-cell stage, which is called as early morula. Okay, this keeps on dividing. You can see the days at one corner. You can see that the morula is going towards the uterine cavity. You can see that. Okay, this is the narrowest portion of the fallopian tube, which is called as isthmus. Okay, which is clinically important. We will discuss why it is important later. You can see that number of cells are increasing. Yes, this is a stage which we have to keep in mind. Now you can see that around 5 to 7 days, the entire morula becomes a different structure. Okay, this is called as blastocyst. What is a cyst? Cyst means a fluid filled cavity is called as a cyst. What is it? Fluid filled cavity is called as a cyst. Now, how does that cavity develop? This blastocyst formation can come as a short note question. Okay, what are short note questions till now? One is cleavage division. Next is morula. Morula and cleavage division usually we will ask during vivas. We don't ask during examinations. Exam short note question is blastula, or sorry, blastocyst. Blastocyst is a short note question. Under that, what you are supposed to write? What is a blastocyst definition and how it is formed? So you will write from cleavage division to till here. What is here? I will tell you now. Okay. Now blastocyst, we will just go a little back. We will try to see this video once again. Now look at it. What is happening? This 8-cell stage, which is called as morula, early morula, it becomes 16-cell stage, 32-cell stage, and finally divides and divides and divides, and it becomes around 108-cell stage. Okay, that is the average number, 108-cell stage. Now when becomes this many number of cells, slowly the uterine fluids, uterus is bulky at this time, it is highly secreting. So what happens during this phase? The secretions of the uterus slowly accumulate around this developing blastocyst, and the fluids will start entering between the cells. Okay, now automatically all the 108 cells will start dividing themselves into two different parts. What are the two different parts? A layer which is outside, okay, and a group of cells which are inside. This particular stage is called as blastocyst. You can see that now, formation of blastocyst. Okay, this is still late morula. After late morula, we will see this blastocyst stage. You can see this now. You can see that a group of cells are there outside. How we are looking at the embryo or the zygote? It's not embryo yet, it is still a zygote. Okay, so how are we looking at the zygote? We are taking a section of it and we are looking inside. If you are looking from outside, it is just a small ball, okay, which is covered by the cells and zona pellucida. Okay, now if you see inside zona pellucida, what are the structures we can see? We can see a big ball of cells which are covering outside. This is called as, this layer is called as outer cell mass. Outer cell mass, I hope you are noting it down somewhere. Okay, this cells, group of cells are called as outer cell mass, or it is also called as trophoblast. What is it called as? Trophoblast. Okay, TROPHO, trophoblast. Okay, BLAST, blast, trophoblast cells. These trophoblast cells are the ones which forms the outer covering. Now only few cells like 8 to 10 cells will accumulate at one corner of this sphere, and they are simply lying on one corner where the uterine fluids just accumulate and push these cells to one corner. Okay, everything is very important. Every single step is important. Now these cells which are inside, these cells are called as inner cell mass. Okay, what are these called as? Inner cell mass, or these are also called as embryoblast. Okay, are you guys following me? Everyone,
Yes, ma'am.
right? So we have got an outer trophoblast and inner embryoblast. Now this cavity is called as blastocele. Blastocele, COELE, blastocele. Okay, this cavity is called as blastocele. Now at this particular stage, we call it as blastocyst. Okay, so blastocyst, you should be writing what is it. You should be writing from which days you will see this formation of blastocyst, and then you will write what is the formation, how it is formed. Then next you will write what are the parts of blastocyst. You will draw a diagram with it, and then you will write what is the fate of the blastocyst. After formation of blastocyst, what is going to happen next? Okay, so this completes your short note question. Clear, everyone? All of you? Guys, please answer.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Right.
Ma'am, can you repeat the steps again?
Steps of blastocyst question, you should be writing define blastocyst. What is definition? Next you will write how it is formed. Okay, formation of it. So you will start writing from cleavage division. Okay, then comes cleavage to morula, early morula, late morula, then blastocyst formation. Then comes the days, which days the blastocyst formation is seen. Okay, then you will write what is the fate of blastocyst formation. Okay, fate, matlab after blastocyst is formed, how it is going to convert, which we will discuss it right now. Okay, then comes parts of a blastocyst. What does it contain? Okay, you can write parts first and then you can write fate, no problem. Then you write applied embryology. We will discuss that applied embryology also. Right, that completes your short note question. Now see here, what is happening here? A blastocyst is formed. The miraculous thing which is happening, you can see 4.5 days to 7 days. Okay, I'm not giving this exact date, 4.5 days. Sometimes till 7th to 9th day, this blastocyst will reach the embryonic cavity. Sorry, uterine cavity. Okay. Now what is happening here? Did you notice the difference? Can you tell me any difference? What did you notice? Anyone?
Increase in
The
blastocyst.
cavity,
Increase in blastocele.
Increase in one by one.
the cavity size increased.
Cavity size is increased. Okay. Anything else?
Zona pellucida is absent and the blastocyst has increased in size.
Excellent. Excellent. This is the answer. Zona pellucida is absent. If you all notice this, we'll just go a bit back side. Now see here. What is happening? You can see the blastocyst cavity. Can you see the difference? The zona pellucida is gone. Okay. This is the beauty of human body. It is accurately designed in that way. You see that? The zona pellucida, what is the importance of zona pellucida? That the zygote which is formed in the fallopian tube will reach the uterine cavity, and then only the zona pellucida goes off. Then you might ask me why zona pellucida is important. Because the trophoblast cells which are present in the outside, they eat up anything whichever they touch. Why they eat up? Because this facilitates the entire embryo to get implanted. Okay, that means if any surface this trophoblast come in contact with, it will start digging up those cells, okay, removing those cells, and it will take the entire embryo inside the wall of the area. Now let's say the zona pellucida is gone in fallopian tube. What can happen? Come up with answers. What can happen?
Implantation.
Yes, implantation can happen in the fallopian tube, and that's called as tubal implantation. Okay, that's called as tubal implantation. Guys, what is this noise which I'm getting? Okay, so you can see that tubal implantation can happen. So the zona pellucida is accurately hatching. How it is, what is happening to zona pellucida? It is not just disappearing, it is hatching like an egg. Okay, the zona pellucida acts like an shell of the egg that hatches out. Excuse me, give me a second. Okay, it hatches out and releases this trophoblast inside the uterine cavity, no place else. Okay, that's the beauty of human body, and it happens so accurately every single time. See here. Now what happens immediately? Now the trophoblast will start growing into number of cells, and it will start eating up the nearby area, whichever place it is. It can be mostly the site of implantation. This is called a site of implantation. This comes your next question. Site of implantation. What is the site of implantation? If this is the entire uterus, this is the uterus, this is the cervix, these are the fallopian tubes, ovaries. Where is the site of fertilization? Fertilization happened here in the ampulla of the fallopian tube. Where is the implantation happening? Implantation happens in the uterine cavity. Are you understanding how you are looking at this picture? We have removed off half of the uterus so that you can see the cavity of the uterus. Are you guys clear?
Yes, ma'am.
Yes, ma'am.
One second. So clear here, everyone? Okay, very quickly we'll go back once again and revise. What is blastocyst formation? From the site of fertilization, that is ampulla of the fallopian tube, zona pellucida still remains intact, and there is division of single cell into two cells. One cell is larger and one cell is smaller. These are called as blastomeres, and every single cell is totipotent. Totipotent means each cell can give rise to an entire embryo. Remember that. So I told you at the beginning, first class of embryology. So cleavage furrows appearing, dividing into number of cells. You can see single cell to 4 cell stage, 8 cell stage. 8 cell stage is called as morula, early morula. Early morula becomes late morula by 16 to 32 cell stage, then finally 108 cell stage, then it starts converting into blastocyst. How? Where the uterine fluids start accumulating inside this blastocyst and pushing some of the cells to the outer coat. This outer coat cells are called as trophoblast, and the cells which are accumulated at one corner, it is called as embryoblast, and the cavity is called as blastocele. Okay, zona pellucida is still intact. By 7th to 9th day, there will be hatching of the zona pellucida. Okay, that hatching is facilitated by uterine environment too. Okay, uterine acidic environment facilitates the hatching of zona pellucida. It gets proteins. Obviously, zona pellucida is made up of proteins, and the zona pellucida proteins will start recognizing the acidic environment of the uterus, and they get a clue that this is the time where my time is finished. So the zona pellucida starts getting removed, and from at that time, the embryo which is inside, the embryoblast which is inside, is hatched out. That is called as blastocyst. A very interesting experiment was done. Okay, which cells, why some cells go for trophoblast and some cells goes for embryoblast? What did they do during this experiment? They took two pronuclei. Okay, pronuclei means this is all laboratory experiments. Okay, in IVF, what do? We take two different cells in a petri dish and we try to fuse them, a sperm with an egg outside in the laboratory, which I'll show you. That video is also there in this presentation. Now, they took any two nuclei can fuse. If I take two female pronuclei and fuse them, they also can fuse, right? Because they are simply they are pronuclei. Okay, they don't have any specific property like, yes, only male pronuclei can fertilize a female pronuclei, two female pronuclei can fuse, or same like that, two male pronuclei can fuse, right? They also can cause fertilization. So what did they do? Experimenters, they tried to fuse two female pronuclei and observe what is happening. Let's see whether it is going to the next stages or not. When they have done this, the cells start dividing, zygote formed, okay, morula formed, and you could see that when two male pronuclei are fused, only trophoblast is forming. Okay, embryoblast is not forming. From trophoblast comes the next stage, that is placenta development. Placenta development is happening, but not the embryo development. Okay, then they decided that yes, only trophoblast can be derived when two male pronuclei are fused. Now they came to the next solution. Let's see two female pronuclei. Okay, maybe there might be a chance. Maybe we might get an embryo from fusing two female pronuclei. When they did that, they have observed that yes, cleavage division started, morula formation started, blastocyst stage formation started, but unfortunately, only embryoblast is developing, not trophoblast. Okay, what is the point if there is no trophoblast? Placenta formation is not happening. Only the fetus is getting developed. But if there is no placenta, the fetus cannot survive. So this is when they have came to a conclusion that yes, male pronuclei gives rise to the formation of trophoblast and related structures like placenta, umbilical cord, everything which supports the fetus from outside. Okay. Whereas when female pronucleus is giving rise to the fetal development. Okay, so both are integral and both are necessary to each other. Okay, see the beauty of our development. One human is developed with fusion of male and female pronuclei and perfectly synced with their both development, right? Yeah. So now let us go to the next stage. This is how electronic microscopic images of formation of blastula. So you can see that This is how cleavage furrows initially start developing, and then slowly they are increasing in sizes, and you can see that how the cells are dividing. Okay. So this is the number of hours and days. You can see that. So this is a picture taken from Langmans. You can see that from 7th to 9th day our implantation completes. Okay, so that completes our first week of development. What is happening here? You can see the brownish red color covering the zona pellucida that is getting off. Okay, and you can see that around 7th day, this is the wall of the uterus. You can see that uterus has got 3 layers, if you still remember: perimetrium, outer covering, myometrium, and endometrium. So implantation is happening only within the endometrium. That's why endometrium is called as stratum functionale, the functional layer of the uterus. All cyclical changes, implantation, development of the baby, everything happens within only endometrium. Okay, the fetus is growing, that is growing within this endometrium. The fetus cannot pass through the myometrium. Myometrium is a muscle, and it stretches to such extent that it wraps the entire fetus without any problem. It can stretch and accommodate the developing fetus. What is the function of myometrium? That is for contraction, that is for expulsion of the products of fertilization, or maybe products of menstruation. Okay, so that is the function of myometrium. All right, are you all clear with this?
Yes,
Yes,
ma'am.
Yes, ma'am.
Okay, till here everyone clear? All of you, first week of development?
yes, ma'am.
Yes.
Now let us see the next video, that is implantation. We don't want this music. So what is happening at implantation? First, step 1, zona pellucida hatching. Then comes the trophoblast. Now see here, oh, 1 second. Sorry. What is happening next? It is going to the site of implantation. What is the site of implantation? This is a very frequently asked MCQ, Viva question. Okay, site of implantation. In implantation short notes, you are supposed to mention this. Okay, site of implantation, side heading should be seen, must be seen in your question. Site of implantation is posterior wall of fundus of uterus. Okay, uterus is divided into 3 parts. We have already discussed that. Fundus is the most dilated portion of the uterus. Then we have body, and we have cervix. Cervix is the lower narrow portion of the uterus. So wherever fundus is there, upper dilated portion, so posterior wall is the most common area where we can see implantation process is taking place. Now it can happen anywhere in the upper segment if you go clinically. That means when you are in a obstetrician or a gynecologist or a radiologist, in fact, who is looking at the site of implantation, you see that uterus is divided into 3 segments: upper segment, middle segment, and a lower segment clinically. Okay, so in upper segment anywhere is okay for implantation. Okay, so if it is going for lower section, I mean the middle section, at the early pregnancy, middle section is very common. Okay, these pregnancies are usually advised to take proper bed rest, because as the embryo is growing, it slowly moves to the upper segment of the uterus. Still they are not at risk, but in the lower segment of the uterus, if there is implantation, it is very, very dangerous for the fetus. Okay, we will discuss that when we are talking about applied embryology. Okay, Now see here, what is happening? This is the upper segment, fundus. So anywhere this portion is a normal area of implantation. If you go a bit lower, this is the middle segment, and the lowest most, that is the lower segment. Now see what is a beautiful change, what is happening. We are starting with second week of development. In second week, the trophoblast, outermost cells, we study changes. From second week onwards, we study changes in two parts. That is changes which are happening in the trophoblast and changes which are happening in the embryoblast. Am I clear till here, everyone?
Yes, ma'am.
Okay, if you got any questions, please note it down and ask me, okay? We have less than a minute. Please join back with the same link. Okay, so we can see that what are the changes in the trophoblast. First, let us understand that, okay? The trophoblast now, these cells, see, however you are looking at the picture, you please understand this is a full sphere, okay? Full circle. How I am showing you, I am taking a section and I am showing you from inside. This is how it looks. There is a cavity, some cells inside, okay? Now, remember that I have cut down half of it. If you are looking from outside, it is a sphere, okay, like a tennis ball, okay? So, or like a golf ball with a lot of pits from outside. So that ball, I'm taking a section and I'm showing you inside. So please remember that whatever pictures you are seeing, these are the sections of the images, okay? They are not complete pictures. Am I clear, everyone?
Yes, ma'am.
Yes, ma'am.
Right, perfect. So now see here, what we are looking at is a section of the blastocyst, the trophoblast, embryoblast, blastocele, okay? Now what is happening? The trophoblast slowly divides and gives rise to number of cells. Which are the number of cells? See here, what is happening? See the beauty of it. Only at one corner we can see huge development of number, okay? These cells which are on this corner,